Our fourth team presentation is in the area of material science and nanoscience. We have Alyssa Chen from Highland Park High School and Shriya Das from Hockaday School, both in Dallas, Texas. Their mentor is Jinming, professor of oncology and pharmacology at UT Dallas. Ladies.
Today, we would like to present our summer research project in which we found a new adjuvant that strongly stimulates immune response. This adjuvant can potentially be used to develop new vaccines for diseases such as HIV and cancer. In the early 1950s, polio affected more than 25,000 people per year in the United States. By 1955, polio was treated more frequently through the use of the polio vaccine. By 1960, the number of reported cases in the United States decreased to 3,000. Through the development of an effective polio vaccine, polio has been fully eradicated in the United States today. This is also true for other diseases, such as smallpox and measles. However, there are many diseases that run rampant through the United States even today, such as HIV, tuberculosis, malaria, cancer, and even influenza, for lack of an adequate vaccine. Many vaccines target T cells and B cells and certain strains, but ignore the potential of the innate immune system. Innate immunity is the body's first line of defense and is not antigen-specific. It functions through the use of antigen-presenting cells, or APCs, and which activates cytotoxic T lymphocytes, or CTLs, as well as helper T cells. Thanks to the work of Dr. Bruce Butler, Ralph Steinman, and Jules Hoffman, we now have a better understanding of the innate immune system. The dendritic cell is an important cell that facilitates innate immunity. The dendritic cell is named for its dendrites, or finger-like projections, which function to engulf foreign substances between the sizes of 20 to 100 nanometers. Once engulfed, the dendritic cell breaks the foreign substance into peptides. These peptides are then processed and combined with major histocompatibility molecules, which are then presented on the surface of the cell, where the T cell receptor binds to this presentation and the CTL is activated. The safest vaccine is the subunit vaccine because it uses purified antigens, which reduces the chances of an adverse reaction. However, the subunit vaccine is not as potent as other vaccines and therefore requires the use of an adjuvant, or a molecule which stimulates interferon and cytokine production. c-di-GMP has recently, a bacterial second messenger, has recently been found to have adjuvant properties. c-di-GMP binds to the endoplasmic reticulum protein called STING, which then activates the protein kinase TBK1, which initiates a signal transduction pathway, which activates transcription factors IRF3 and NF-kappa B, which leads to the production of type 1 interferons, which is important for the activation of T cells and B cells. While c-di-GMP alone is adequate, its inability to penetrate through the plasma membrane of cells limits its effectiveness because only a fraction of the c-di-GMP exposed to the environment will be engulfed by a DC. Thus, we propose to encapsulate the c-di-GMP into a nanoparticle between the sizes of 20 to 100 nanometers, which are the sizes that DCs are most prone to engulf. This led to an internship at Dr. Jinming Gao's lab, which specializes in the development of nanoparticles. Our nanoparticle used was pH-tunable and micelle-based. Micelle-based means that the nanoparticle had a hydrophobic core and a hydrophilic shell. pH-tunable means that our micelle had a specific pH transition point. For our purposes, the nanoparticle will be called UPS 6.9, or ultra pH sensitive, with the transition point at 6.9. This means that above the pH of 6.9, our nanoparticle was a micelle, But below the pH of 6.9, the nanoparticle became protonated and dissociated, releasing the c-di-GMP inside. Our nanoparticle would be delivered through a process called endocytosis, where a vesicle would be formed, then an endosome, and later a lysosome. Throughout this process, the pH would decrease, which we hoped would allow our nanoparticle to dissociate and release the c-di-GMP inside. We approached our experiment with the theory in mind that by directly delivering the nanoparticle to the DCs, this would increase the yield of the c-di-GMP and thus heighten the immune response. The hallmark markers for DCs are CD11C and MHC2. In order for the innate immune response to be activated, two steps must occur. First, antigen presentation, which is where once the peptides are processed and combined with the MHC molecules, they are presented on the surface of the DCs where a CTL can bind to this presentation. Secondly, co-stimulation must occur, which is where a co-stimulatory molecule on both the DC and the T cell must bind. Thus, we tested for two markers. First, MHC1, which is an antigen presentation marker, and secondly, CD86, which is a prevalent co-stimulatory molecule. An increased expression of these two molecules would suggest an increased immune response.
We began our experiment by first creating our UPS 6.9 nanoparticles through a process called sonication, which is basically using sound energy to rapidly and evenly disperse the polymers to form uniform micelle-based nanoparticles. We created two sets of these nanoparticles, one with encapsulated c-di-GMP, as shown here, and another with no load. Our second step was to culture DCs. We obtained bone marrow left over from some graduate students who used the bone marrow for previous experiments. We then stimulated and cultured this bone marrow to develop into conventional DCs, and then used these cells to plate 10 wells in a 96-well plate. We then performed two trials testing the effect of 5 conditions on these cells. These 5 conditions include poly-IC, which acts as our positive control, since it has been previously proven to stimulate an immune response. Our independent variable, UPS 6.9 with encapsulated c-di-GMP, and our negative controls, which include empty UPS 6.9, free c-di-GMP, and no treatment. Following treatment, we stored the plate in a 37 degrees Celsius incubator overnight, and the following morning, prepared and stained these cells for fluorescence-activated cell sorting analysis, or FACS analysis, and then used the computer program FlowJo to analyze our data. Now again, FACS stands for fluorescence-activated cell sorting, and basically the purpose of FACS is to analyze cells. It's to separate cells based on their level of cell surface marker expression. In order to analyze through FACS, we had to stain our cells with these fluorophore conjugated antibodies. Specific antibodies bind to their corresponding markers. So if a fluorophore is conjugated onto an antibody, which is bound to its corresponding marker, then essentially that fluorophore represents this marker. In the FACS caliber machine, a laser is emitted, and when that laser hits the fluorophore will emit a light signal. Each fluorophore emits a light signal at a different wavelength, which allows us to differentiate between the markers that they correspond with. Because of this, there is a direct relationship between light intensity and expression level. The higher the light intensity, the greater the cell surface marker expression. This graph portrays the format of our data. The x-axis represents the light intensity, while the y-axis represents the percent of the maximum number of cells that have emitted a light signal. Since the shift to the right in the graph represents an increased CD86 expression, in this graph it is clear that cells stimulated with the UPS 6.9 nanoparticle with encapsulated c-di-GMP, shown here in red, experienced a greater CD86 expression than when compared with cells stimulated with our positive control, poly-IC, shown here in blue. A similar pattern follows when compared with cells stimulated with free c-di-GMP, shown here in green, as well as when compared with cells stimulated with an empty nanoparticle, shown here in light blue. Similarly, a shift to the right in the graph represents an increased MHC1 expression. So in this graph, cells stimulated with the encapsulated nanoparticle, shown here in red, experienced a greater MHC1 expression than when compared with cells stimulated with our positive control, shown here in blue, as well as when compared with cells stimulated with free c-di-GMP, shown here in green, as well as when compared with cells stimulated with the empty nanoparticles, shown here in light blue. In conclusion, our experiment demonstrates that cells stimulated with the UPS 6.9 nanoparticle with encapsulated c-di-GMP experiences greater MHC1 and CD86 expression. To summarize, when a DC is exposed to a nanoparticle, the DC will engulf the nanoparticle, subsequently forming an endosome. And as the pH in this endosome decreases, the nanoparticle will dissociate, releasing the c-di-GMP. And the endosome will then release the c-di-GMP into the cytoplasm, where it can then bind to the STING protein, initiate a signal transduction pathway, and activate transcription factors, which lead to the production of interferons. Interferons upregulate the production of co-stimulatory and MHC molecules, and upregulation of these molecules activates DCs and therefore cytotoxic T cells. We demonstrated that our nanoparticle can activate DCs. However, this is only one of the important steps necessary in developing an effective vaccine. In the future, we should test whether our nanoparticle can activate helper T cells and cytotoxic T cells in vitro and in vivo in mice. In addition, we should test whether our nanoparticle with encapsulated c-di-GMP and an antigen specific to a disease can become a potent adjuvant to develop more effective and safe vaccines for the treatment of infectious diseases and cancer. We would like to thank our mentors, Dr. Jinming, Dr. Wang Hua, Dr. Gan Hua, and Dr. Zijian Chen, for mentoring us and allowing us to use their facilities this summer. We would also like to thank the Siemens Foundation, College Board, and the George Washington University for providing us this opportunity and for hosting us this weekend. Finally, we would like to thank the audience and the judges for your time today. Thank you.